As EMSL Director, I have the privilege of introducing the recipient of the 2019 M.T. Thomas Award for Outstanding Postdoctoral Achievement. This award recognizes outstanding accomplishments by a postdoctoral research associate who has performed at least a portion of their research at EMSL. I'm pleased to announce Dr. Dusan Velickovic as this year's recipient. Congratulations, Dusan. Dusan is the 24th scientist to receive this award since its inception in 1996. As part of the process, EMSL's User Executive Committee served as a selection board reviewing the nomination packages. I'd like to thank the User Executive Committee for reviewing and selecting this year's award recipient. The M.T. Thomas Award is named after Dr. M. Tom Thomas, a distinguished Pacific Northwest National Laboratory scientist who was part of the leadership team starting in 1987, and he retired in 1995 as EMSL's first operations manager. The M.T. Thomas Award recognizes a postdoctoral researcher who has used EMSL resources within the last 3 years to develop a conceptual advance that has impacted or the potential to impact their field. To be eligible for this award, the candidate must demonstrate a high level of scientific accomplishment, creativity, and problem solving relevant to the EMSL mission. Dusan met and greatly exceeded these criteria. Dr. Chris Anderton, EMSL's biogeochemical transformations team lead, was Dusan's mentor and nominated him for this year's award. In the nomination letter, Chris said of Dusan, "I have been fortunate to work with many bright scientists through my short career, and Dr. Velickovic is by one of the far most creative, imaginative, and driven young scientists I have worked with." Dusan started his outstanding postdoctoral career at EMSL in 2016, and in 2020, he became an EMSL scientist. His research is focused on biological mass spectrometry, specifically matrix-assisted laser desorption ionization, or MALDI, mass spectrometry imaging. As a postdoc, Dusan transformed our mass spectrometry imaging capabilities and expanded the number of EMSL users interested in using mass spectrometry imaging from more than a few to a half dozen per year. Dusan's efforts also have led to an increase in external funding for mass spectrometry imaging to support important biological research. One of Dusan's many achievements was his research with EMSL user Gary Stacy at the University of Missouri to understand metabolic transactions occurring in plant microbe interactions. This work was highlighted in the Department of Energy's budget request to Congress for fiscal year 2020. Some of Dusan's other accomplishments as an EMSL postdoc included co-authoring 12 scientific publications in peer-reviewed journals, 8 as first author, delivering 8 conference presentations, 2 invited, 2 oral, and 4 posters, supporting 10 user projects resulting in 7 publications, winning 2 travel awards, and co-authoring 3 external and 2 internal proposals, 1 funded and 1 pending. Chris summarized why Dusan should receive the M.T. Thomas Award by saying, "Dr. Velickovic has an entire body of work that has made a broad impact across EMSL and the field of plant biology." A native of Belgrade, Serbia, Dusan earned a doctorate in chemistry, analytical chemistry from the University of Belgrade when he graduated in 2011. He was the youngest PhD candidate in the biochemistry department at the time. As this year's M.T. Thomas Award recipient, Dusan will receive a plaque recognizing him for his outstanding postdoctoral achievement. He will also receive a stipend of up to $2,000 to attend a domestic conference of his choice to serve as an EMSL ambassador and present his research using EMSL science capabilities. Dusan, congratulations again. Thank you for all your contributions to EMSL and the scientific community. And now I'd like to invite Dusan to come and share some of his research that he conducted while at EMSL. The topic of his presentation is molecular snapshots of complex environmental biochemistry. Please welcome Dusan.
So thanks, Douglas, for this really kind introduction. And of course, thanks EMSL User Committee for selecting me for this this year M.T. Thomas Award. It's really my great pleasure and honor. As Douglas said, today I'm going to talk how we can use actually molecular imaging to better understand complex environmental biochemistry. But before I dive deeper into this topic, let me also give big thanks to my mentor, Dr. Christopher Anderton, who supported my application for this award, supported my entire postdoctoral career, and definitely without him, all those results that I'm going to present today would not be possible. So thanks, Chris. And also I would like to thank 2 really valuable EMSL users, Gary Stacy and David Weston. They also supported my application for this award, and most of result I will present today actually resulted from their EMSL user projects. So this is a short outline of my talk. As you can see at the beginning, I will give just brief introduction about mass spectrometry, molecular imaging, principles, tools, why this is important, and then how in EMSL we actually adapted this tool that we can better understand different biological environmental questions. And finally, I will close this talk with some challenges and future directions where I see that environmental science can additionally benefit from molecular mass spec imaging. So mass spec imaging is actually spatial probing of sample surface chemistry by mass spectrometer. Idea is that basically we go from spot to spot in the sample. So from each spot of the sample, we acquire mass spectrum, and that mass spectrum basically reflects molecular composition of that particular area that we imaged. So that we call this area that we image, we call pixels, and that size of pixel can be from several cells to single cell, depending what probe we use, how well we can focus our probe. Idea is that when we finish entire sample surface scanning, we are able to create and reconstruct molecular ion images that basically reconstruct from mass spectrum, so that those ion images basically represent relative molecular abundance over sample surface molecules that have particular m over z values or mass, if you will. So there are numerous ways how we can do sample surface sample probing. And I will now go deeper now in explanation each of them. I want to highlight that all of them differ in mass resolution we can achieve, in molecular information we can obtain from them. But one technique that I really want to highlight here is probably the most common for molecular mass spectrometry imaging. So for imaging of intact molecules is matrix-assisted laser desorption ionization, or MALDI MSI. This technique uses basically compounds that we call the MALDI matrix. So those compounds are usually small aromatic organic acids or bases that are added in our sample and basically enhance and help in ionization of endogenous molecules by absorbing basically UV energy of MALDI laser that we use as a sample probe. Okay, now it would be completely unfair toward this technique if I immediately jump how we adapted in EMSL this tool to address biological environmental science, if I don't say that really the greatest application of this technique is in clinical research. So reasons are obvious. We can use this technique to locate and identify tumor markers, to identify drug targets, or for example, we can use them as we can use this technique as really high throughput technique for tissue classification studies. Okay, and now I want to show you how in EMSL we adapted this tool to address several biological environmental questions. And the first example that I want to present today is process of biological nitrogen fixation. So this biochemical reaction actually occurred in the specific organs on the root that are called nodules. They're characteristic for leguminous plants. And those organs are basically symbiotic organs between microbial cells that infected plant root cells. So those microbial cells basically fix atmospheric nitrogen, and then that fixed atmospheric nitrogen is assimilated into plant biomolecules. And in the opposite direction, plant provides energy for this really high energetically expensive process. So we can see that this is really tightly metabolically regulated system, and intense metabolic exchange occurred to sustain efficient biological nitrogen fixation. Our molecular imaging results, especially 3D imaging data, show that although most of molecules of metabolites show spatial compartmentalization. Actually, they are present either in infection zone, so this central zone of nodule, either in inner zone or outer cortex. Some of the molecules show actually asymmetrical distribution throughout the infection zone. Example is, for example, S-adenosyl methionine, and this completely contradicts to long-standing hypothesis that this infection zone of soybean root nodule is actually metabolically uniform, homogeneous compartment, and value of this we can see if we just see role of S-adenosyl methionine in the downstream of biological nitrogen fixation. For example, S-adenosyl methionine is central molecule in biosynthesis of polyamines, molecules that serve, for example, as growth, soybean root nodule growth elongation factors, or for example, in biosynthesis of phosphatidylcholines, actually molecules that are involved in recognition between bacteria and plant cells. Second example that I want to highlight here actually is another symbiotic system, in this case between ectomycorrhiza fungi and roots. So this process called ectomycorrhiza formation, and here again fungi serves to improve fitness of plant and nutrition of plant in exchange to plant carbon. Again, this ectomycorrhiza formation is tightly regulated process by biomolecular signaling between plants and fungi, and secondary metabolites play a really important role in this communication. However, one class of molecules that was not previously acknowledged as important for recognition and this interaction were alkaloids. And alkaloids are, all the alkaloids are really ubiquitous in plants, in pine roots, for example. They are well rich in piperidin alkaloids, and they serve as nitrogen containers and also nitrogen reservoirs, and also like protecting molecules for plants. Our data, however, our molecular imaging data show completely different alkaloid profile in pairings of fungi and roots that are compatible. Actually, in those compatible pairings will induce morphological changes in the hyphae and in the roots, while in incompatible pairings, those changes will not occur. And we show that alkaloid profiles are completely different. And also from our imaging data, we also can see that plants relocate alkaloids during formation of ectomycorrhiza, so from a very root tip to almost entire root. How we can, because here we can observe from early to late how advanced this accumulation of those alkaloids. And those data indicate that those alkaloids actually might be responsible for that host fungi specificity. Third example that I want to show you today is a peatland ecosystem, and this model system is probably the most utilized in our mass spec imaging lab, not only for addressing biological questions such as, for example, in this case, cycling of carbon, but also for developing and for benchmarking our mass spec imaging tools. So those peatland ecosystems are composed from peat moss that associate with cyanobacteria and with fungi. And so we can see that here we basically have 3 kingdoms, and we can grow them in the simulated conditions. So in the agar petri dish, basically we can separate them in space and then use our mass spec imaging tools to describe the molecular communication between those species in a tripartite community. Okay, before I go into results results related with this project, I want also to introduce another mass spec imaging tool, actually probing tool in our lab. It is liquid extraction surface analysis, or LESA, comparing just reminder, so we use, in LESA, we use droplets, or like we perform some kind of micro extraction. We use the droplets for micro extracting the sample surface, and comparing to MALDI, this technique provides actually quite poor spatial resolution. In MALDI, we speak about, let's say, 20, 30 micrometer scale. In LESA, we speak more about 200, 300 micrometer scale, because we use really droplets to make those micro extraction. However, LESA has advantage comparing to MALDI, that we can relatively easily couple it to instruments and that will make available actually to perform in untargeted fashion, for example, hundreds of MS scans from single pixel. And also we can couple it, for example, with ion mobility device quite easily to provide us, again, in untargeted fashion, information about ion mobility of samples, about ion shape of the samples of molecules in each pixel that we analyze. So basically, LESA can provide us more information about molecules that we image, not only M over Z value. So exactly that approach, so coupling LESA to Orbitrap and then performing MS analysis on hundreds of molecules in a single pixel, enable us actually some really great insight into metabolic pathways. For example, we were able to separate, and not to separate, but to map two different metabolic pathways that occur in different parts in different species of this community, but common for them is that they share same molecules with the same molecular formula. We call those molecules mass isomers, and without any orthogonal analysis, we would not able, for example, to discriminate between 3-methyl-lysine and the diamino knot that are basically mass isomers. In our case, we were able to show that they are located in different species of tripartite community, one in cyanobacteria, one in fungi, and you can see that, for example, 3-methyl-lysine is a precursor for carnitine biosynthesis, while diamino knot is precursor for biotinamide biosynthesis, two molecules that are really important for fatty acid metabolism. However, this approach can only provide us general information about regio and stereoisomers, because regio and stereoisomers have very similar, almost undistinguishable MS patterns. So basically, what we can conclude, for example, is that in fungi, we can see, for example, alpha anomeric sugars, or in cyanobacteria, 1,4 or 1,6 glycosidic bond disaccharides. But what is identity of those disaccharides, we cannot really tell by our approach. Also, another risk that we have here, because we perform that micro extraction and then that droplet we insert directly into the mass spec. We don't have any chromatographic separation before our fragmentation. So actually we have that risk that several isomers can be in single sampling area, and then that we basically fragmented a mixture of mass isomers. So drawing of conclusion can be quite ambiguous. And it is a reason why we advanced our technology and coupled the liquid extraction surface analysis with ion mobility device, in this case SLIM, so structure for lossless ion manipulations. And this enables us that basically in each pixel we can separate those disaccharides according to their shape, or according to their collisional cross-section, if you will. And by using internal standard, we were able basically to visualize in each part of community what contribution is of each disaccharide. And basically these results completely change our understanding and our former hypothesis that sucrose actually is the main disaccharide that is that serves in interaction between those communities. We actually show that trehalose, as you can see, is present in each part of this community, especially in the moss, which was quite interesting. And this completely changed our understanding from of trehalose as a relatively obscure molecule in plants, one that actually may have a really crucial role in that carbon cycling in peatland ecosystems. Final example that I want to show here is actually not resulted of EMSL user project, but from Jim Moraine early career project. But this one can be really easily applied to our EMSL users. And it is how we can actually bypass challenges in attaining direct molecular, untargeted molecular information from rhizosphere, from root soil interface. You know, all those challenges are usually related with basically porosity of the soil and our inability to prepare it well for direct untargeted molecular imaging. Here we propose way that basically we can use membranes to spatially transfer molecules from rhizosphere on those membranes, and then that basically we can image indirect those membranes by our MALDI or some other MSI tool. And as you can see, we revealed that basically we can image really a lot of different organics in the rhizosphere. We can map them in different locations in the rhizosphere. Some of them are co-localized with the root itself. Some of them are present in specific pockets in the rhizosphere. Identity of this molecule revealed that some of them are also root exudates. Some of them are basically product of metabolic, of microbial activity. So this is a really great way to go beyond biochemics analysis that are currently available and to provide some microscale chemical heterogeneity inside the rhizosphere. And also great advantage of this technology is that basically we can provide temporal mapping of for example, of growing a root or developing of the rhizosphere, because we can every time change and use the fresh membrane, and that way we can basically map how growing or elongating root influence its microenvironment. However, we are still not perfect. We are still not yet there. There's still a way that we need to improve. One of the most important things is that we need to preserve native morphology of the sample better, so that we can basically much more accurate describe origin of molecules that we image, so that we know from which cells those molecules arise. Also, we need to make this technique more quantitative. So not just saying here we have more, here we have less, or here we have 2 times more or 3 times less. We want to do it in untargeted fashion to cover as much molecules as possible in quantitative way. And also probably the most challenging task will be to increase, we call it sensitivity, but actually to increase coverage of metabolites that we can see and that we can map in samples, either in the tissue samples or in the rhizosphere itself, to be able to cover as much as possible molecules. We need, of course, here are listed some examples that we need to find better matrices. We need to increase ionization efficiency. We already have tool in EMSL in terms of MALDI-2 capability. We need also additional orthogonal separation. And with that, I would like to thank you for your attention. And yeah, I'm open for questions. Thanks.